First of all, it's really, really nice to be here, and I also would like to keep this as interactive as possible. So just stop me. We can, sort of even go a little bit Socratical here about the material that I wanted to show you today. So normally we are interested in the lab in understanding molecularly how we get in our brain the many different classes, in our cortex I should say, the many different classes of neurons that we have. What are the molecular rules? What is the logic that builds neuronal diversity there? And also frankly, once we get all the neurons that we have, how do we keep the identity of those neurons unchanged for our lifespan? What are the mechanisms that allow neurons to stay the same for many, many years? And so in a symposium in a couple of days, I'll mostly talk about stability of neuronal identity and reprogramming. What I wanted to do today was to tell you about an approach that we have taken recently to begin to understand this molecular logic that builds neuronal diversity using a high-throughput molecular mapping approach that allows to take now a systems-level approach to understand how many different classes of cells may co-develop molecularly in the same brain. So if you can lower maybe the lights in the front, because we have lost the colors from this presentation, so you have to use a little bit of your imagination here. There were a lot of colors. So the part of the brain, as I mentioned, that we study is the cerebral cortex. You all know that, sorry, I'll go back here. You all know that the cortex, thank you, the cortex controls for some of the highest level function that we are capable of, especially as human beings. And basically, I'll begin to use this thing well. All right. Basically, all of these higher order functions are executed, as Gord was mentioning before, by two broad populations of cells. The excitatory long-distance projection neurons, which basically constitute the entirety of the output circuitry from the cerebral cortex, and the local inhibitory interneurons that Gord told us about, which integrate with the excitatory neuron, and these, of course, need to make this very precise circuit in order for the cortex to really function well. So now a lot of questions remain unanswered about how the brain makes this incredible diversity of neuronal subtypes, what goes wrong when these neurons don't wire together properly, like in the context of neurodevelopmental diseases, and also, as I mentioned before, how do we keep these identities unchanged for the lifespan of the organisms with time? So just very briefly recapping maybe a little bit of what Gord told us, both the projection neurons and the interneurons of the cerebral cortex are only born during embryonic development, contrary to what Alejandro was telling us for the dentate gyros. All of the neurons of the cortex are only made in the embryo. And projection neurons and interneurons are made from two completely distinct sets of progenitors. The excitatory projection neurons are made from progenitors located in the dorsal telencephalon, also shown here, through a span basically of development, and then they migrate radially to populate the layers of the cortex, while the interneurons derive from ventral structures like mostly the MGE, the CGE, and the POA, and then they migrate into the cortex, first distributing tangentially and then invading the cortical plate. And so when neurogenesis is over, then you have a nicely layered cortex, at least in mammals, with 6 layers that are populated by different classes of neurons. All right. It's sort of a never-ending complexity because, of course, things are much more complex than this. The cortex also has many different areas. I'm just showing you here some for the mouse. It gets much more complicated, of course, in humans. And these, of course, process information from different sensory organs. And then if you look under each one of these areas, the neurons are organized into columns of neurons that are interconnected with each other. And the columns are then subdivided into the layers that I was telling you before. And each layer contains many different classes of excitatory neurons as well as many different classes of interneurons. And I don't want to go too much into classifying this because I completely agree with Gord that there is a sort of a utility in classifying cell types. Maybe this is to answer the question that someone asked at the end of Gord's talk as well, to give my 2 cents. I think it's important to classify. It roots things really well. It's really satisfying to be able to call a cell based on a combination of parameters that define that cell type. But when that exercise just becomes an exercise of being able to name every single cell, I think that's not useful anymore. And it doesn't tell you much about how the brain really works. So with that kind of philosophy, I will just, let me see, maybe classify or describe 3 broad group of cells, because these are the kind of cells that we have purified in high throughput and molecularly profiled with this new approach that I'll tell you about in a second. So very early during corticogenesis in a mouse, around E12.5, many different classes of corticothalamic neurons are born. There are many different types. They are all located in layer 6, and they connect to different nuclei of the thalamus. If you wait about a day in mice, then many classes of subcerebral projection neurons are born. So these are located in different areas of layer 5, in layer 5B, exactly. And then they send long-distance axons to a whole set of targets, including the spinal cord. These are the corticospinal neurons, which are below, all these targets are below the cerebrum, so subcerebral neurons. And often people talk about these neurons plus these neurons as corticofugal because their axons go far away from the cell body located in the cortex, so away from the cortex. And then the cortex also, of course, contains many different types of commissural neurons. And these are neurons that connect the 2 sides of the cortex via this large commissure, the corpus callosum. They are present basically in every layer, but a large number of them is born late during neurogenesis. And these are the callosal projection neurons located in the upper layer 2-3. They connect via the corpus callosum, and like other callosal neuron types, they also have a very complex pattern of collateral axons that connect to the striatum, ipsilaterally, contralaterally, and so on and so forth. So, and to the frontal cortex. So again, let's not classify too much, but just remember that these are very complex heterogeneous populations of neurons. So, we deal with this problem that the cortex is full of different classes of neurons, different types. We have known about their morphology, their connectivity, and some of their features for a very long time. Yes? Microphone. Okay, I need to coordinate. I don't have a pocket. For a very long time. But if we really would like to understand what is the molecular logic that builds these different classes of neurons during development, then we face a fundamental problem, the fact that you need to be able to purify these different classes of cells from the cortex in order to study them in isolation and profile them molecularly. If you don't do that, and you just take a chunk of cortex, for example, extract the RNA and do RNA sequencing, you quench every difference in gene expression levels of any given gene across populations, and you're not going to be able to detect these genes. And so how do you purify different classes of cells if you don't have in the first place the markers that genetically allow you to label the cells? And so this is almost an historic slide for me. It's many years ago, but basically a long time ago, we took an approach that today I would say was painfully low throughput. Nobody should do this today to isolate cell types, because this was an approach where we basically use surgery to inject fluorescently labeled microspheres in the terminal targets of the projection neuron types. So in this case, for example, the corticospinal neuron, we would inject the beads in the spinal cord. You would have wait for the beads to be transported back up to the cell body of the neuron in the cortex. And then if you look at the cortex now, what you see is that only corticospinal neuron in layer 5 would be labeled. This is nice, but it takes forever. You basically have to do surgery every time you want to purify a specific class of neurons, and also surgery doesn't give you really the specificity of labeling, for example, distinct population within a broader set of neurons. And we did it across 2 cell types at that time, the callosal neurons with injection in the contralateral hemisphere, and the corticospinal neurons with injection in the spinal cord. And then we put the cells through FACS sorting, which at that time was kind of difficult. Now it's more of a routine procedure. But you can take these labeled cells and put them through a FACS sorter, and now finally you have a pure population of cells 2 years later. So nobody should do this today. I keep saying that. But the study was useful because it allowed us to identify the first set of markers. So now we are at genes that label specific classes of neurons. And here I'd like to highlight one other concept, that no single marker identifies a single population of neurons, especially in the cerebral cortex. Every time I talk about markers, I mean combinations of genes. It's never one gene, it's always combinatorial, and it's always dynamic, meaning that if I take a corticospinal neuron and I think about the molecular code that labels it at E15.5 will be very different from the molecular code that labels it at P14, for example. Not completely different, but different. So time is important, and combinations of genes is also very important. Yes?
This is the group for the cerebral cortex or for the whole brain?
I think it's everywhere. The cortex is particular because the diversity is higher. So there are many more classes of neurons, I think, here, and we don't even understand what they do functionally for a lot of these, and the classification is clearly incomplete. So there are more different types than more basal structures, but I think the same applies to other neurons as well. One gene will never identify in the central nervous system one neuron. Okay. Although this is used through publication all the time. One neuron, one gene, right? And that's really problematic, I think. All right, so what these sort of low throughput approach also allowed us to identify, and I'm summarizing 7 years of work in a single slide. This is work done by Simona Lodato in my lab. It also gave us those transcription factors that Gord was talking about now for the excitatory neurons. One of them was this gene called FASF2, which then with a lot of work found that is a selector gene. So sometimes you get lucky. Sometimes you get transcription factors that alone, and this is one element of the molecular logic, alone we found can activate directly. So by binding directly to the promoter regions of downstream genes, they can activate not one gene, not two genes, but many, 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 many genes. In fact, 80 to 100 genes that are specifically expressed in one population of neurons. In this case, were the corticospinal motor neurons. And then what we found is that these genes, the effector genes, either alone or in combination, will go and define the other features that collectively describe this neuron as a corticospinal neuron. So and here is another principle. No single feature describes a neuron. So we can classify neurons based on single genes. We cannot even classify neurons based on combination of genes alone. You have to begin to combine multiple features to really understand what a neuron is. And I'll give you an example. So in this case here I put molecular features. These are the combinations of genes. But where the axons, for example, of these neurons go, it's also a defining feature of their identity. In fact, the name of these neurons come from where the axon would go. Okay? What kind of neurotransmitter these neurons use is also an element of that classification. The intrinsic electrophysiological property is another element, and so on and so forth. And so in this case, we got lucky because at least co-regulation of gene expression downstream of a single selector gene is important for the acquisition of all of these features by corticospinal neuron. This is nice. But this is an exception. I don't think that this is the rule that will be applied to every single neuron of the cortex because you will run out of transcription factor very, very easily. So it's going to be much more complex and much more combinatorial, but sometimes you get lucky and you find basically these genes. So now I started sort of the presentation by saying that we're interested in understanding the logic that builds neuronal diversity, not single neurons, not single genes, beyond single gene, beyond single neuron. How do we get all the neurons that we have basically in the brain? And so I would argue if that is the goal, we need to take a completely different approach to profiling this diversity, and we need a high throughput approach that allows you to take out different types of neurons from the same brain in high throughput. So no surgery, and I would say not even genetic labels, because I told you that each single neuron can only be identified by combination of genes, and so you run out of genetic driver pretty quickly. After you use your Cree, your Flippy, and maybe a couple of other recombinases that are out there, you cannot go more combinatorial than that. And so a few years ago, we thought that it would be fantastic if we could first define a method that would allow us to label projection neuron subtypes by, first of all, at very early stages of development, when these lineage bifurcation decisions that control the acquisition of identity are made, and before they send axon to any distant target that would allow you to retrogradely label from the target. Okay? So we need to get them when they're very, very early, mitotic. We need to use combinations of markers. So beyond individual genes, we need to be able to use many, many genes, 2, 3, 4, 10, whatever it takes to identify that specific class of neurons. We need to have a method that is scalable, because this has to go in high throughput really quickly for many mice across many different neuron types, and also to allow the isolation of a large number of neurons, because so far we've only talked about transcription, and transcription is one important component of how neurons are made, but we haven't even begun to talk about genetics and epigenetics and proteomics, which requires a very large number of neurons, which is prohibitive at this moment in the central nervous system. And since we're at it, let's do it without genetic label. No generation of mice, no combination of lines, no more expensive mice bills, I would say, but also most importantly, a method that would allow you to purify the cells from tissues and from organisms that cannot be genetically labeled, like, for example, the human brain. And so, several years ago, Brad Molyneux, who I had been working with for many, many years, and he did a brief postdoc in my lab, together with Loyal Goff from John Rind's lab, decided to develop exactly this approach. And we got a lot of help from Doug Melton's lab, who was developing a similar system for the pancreas. So it works across different organ systems. And I'll go and walk you through the steps precisely, but here is the idea. The idea is that one would take a brain and be able to label individual types of neurons within that brain, sorry, this is washed out in the slide, by using combination of genes, combination of antibodies, I should say, that are directed not against surface antigens or neurons, but against transcription factors that are present within the nucleus of that neuron. And because we know the codes of transcription factors that define each individual neuronal subtype to a certain extent, now you can choose your antibody against different factors to identify different cells within the same sample. And we apply these methods, again, I'll tell you the details in a second, to 3 classes of neurons. The callosal projection neurons that connect the 2 hemispheres of the brain. The subcerebral projection neurons, including the corticospinal neurons that connect the cortex to the spinal cord, and the corticothalamic and subplate neurons that connect the corticothalamic, the thalamus. And we did it over this range of development. So the very, very early stages of phase specification, as soon as the neurons came out of the cell cycle and became postmitotic through basically birth, which was a window of time during development that we were never able to catch, because this is a time where the neurons have barely begun to extend axons towards their target that cannot be purified in other manner. So let's look at the details of that. You take a brain, unlabeled, so genetically you don't have to do anything to this brain. So imagine that here I drew a brain, and you think about the classes of neurons that you want to purify. This works not only for the projection neurons, but for any neuronal subtype. And then you think about a combination of transcription factor that would distinguish, for example, these 3 classes of neurons from each other. We knew from our prior work, in our case, that the combination of 3 transcription factors would do that. Only 3, because this is a method that can only detect levels of expression of transcription factors. So if a neuron expresses transcription factor A, if 2 neurons, express the same transcription factor A, but one expresses it at a higher level than the other, these methods detect it. And so the 2 neurons would be differently labeled, okay? Then the brain is, the cortex is taken, is dissociated to a single cell suspension, and there are ways to do that without affecting the well-being of the neurons. Then the cells, and this is important, they're lightly fixed and treated with saponin. And what saponin does, makes little holes in the membrane of the neuron without having the cell exploding, because now the cell is fixed. And this fixation will become important in just a second. Then one adds the antibodies to that vial. And then this entire set of cells, it's put through a fax sorter. So the limit here is only the number of colors that your fax sorter can read. But if you have worked with any immunologist, that's a super high number of colors. You can combine as many antibodies basically as you want here. And then from the same basically set of unlabeled brain, you can purify these 3 classes of neurons. So for example, callosal neurons are defined as BCL11B low, TLE4 low, SAB2 high. But the subcerebral neurons are BCL11B high, TLE4 low, SAB2 low. This sounds a lot like the immune system, where people have been able to purify different classes of cells from the blood based on differential expression of different antigens. So these are the plots. So you can see then for the first time we were able to distinguish the subcerebral projection neuron from the corticothalamic projection neurons very early on during development, basically a day and a half after birth of the neuron itself, and then see them separate from each other molecularly over time. These are the images. It might be difficult for you to see, but the arrow here points at cells that are expressing a certain transcription factor at a very high level, and cells that express it at a very low level. So you can distinguish level of expression of that gene. And then, despite the fact that these cells were fixed. And the fixation is important because now forget about the difficulty of sorting fresh neurons through a FACS sorter, which is not trivial. The neurons get exotomized and retomized. They tend to lyse and are not available for profiling. Here now the cells are fixed. So you basically sort brain like you would sort blood. A very high speed, a very high pressure. There is no problem anymore, and in very large amounts, because you don't have to genetically label the cells before you reduce them to a single cell suspension. And then what you do, you spend a little bit of time, but we have this protocol, and we're very happy to share it with you, to reverse cross-link the RNA from the protein, which happens when you fix the tissue. So this is now possible. And then we made this method compatible with RNA sequencing. So you can sequence actually really, really deep, despite the fact that the cell had been previously fixed. So you can see how we can go really automatically, really in high throughput, all the way from unlabeled brain to RNA sequencing. And then you look at what you get from the RNA sequencing data, and you get this. Let me put it all here. So, of course, you get a lot of genes that are differentially expressed and that we expect that we do have a lot of mRNAs that follow these rules. But the reality is that you now begin to see a lot of transcriptional dynamics that go on as these neurons choose their fate. We didn't even know were there before, like promoter switching, CDS switching, isoform switching, all kinds of things that in other tissue people have studied really deeply, but in the central nervous system, because of this heterogeneity of cells, was very, very hard to detect. I'll give you an example of an isoform switching here. So if you look at this gene here, and if you look at the entirety of the transcript for this gene, you'll see that in all of the different cell types, you have transcript that grow over time. So it's time on the x-axis. But the reality is that there are 4 different isoforms of this mRNA. And if you look at how these isoforms behave over time, it's completely different. So the same transcript produces different kind of isoforms which follow a different transcriptional dynamic and regulation over time. And this is not surprising because we're talking of the cerebral cortex after all, the most complex tissue of them all. And so there must be a lot of regulatory mechanisms here that we haven't really begun to understand that allow the cortex to generate the diversity of neurons that we see there. Okay. This is just an example of some of the genes that we detected that are very specific, for example, to callosal neurons, to subcerebral neurons, to corticothalamic neurons. We went from having about 30 to 40 to 60 markers for each individual lines to having about 700 markers for each one of these neuronal subtypes. And what I wanted to point at is that while some of these are mRNAs, many of them are link RNAs. So link RNAs are this interesting form of RNA. It looks a lot like an mRNA, has a poly A, has exons and introns, and so on and so forth. But it's not translated into a protein. And it's beginning to emerge a pattern by which these link RNAs are actually very important regulators of both chromatin modifiers and help fate selections in a lot of different tissues. The key of these link RNAs is that usually they're expressed at very low levels, and so we couldn't quite detect many of them in the brain before. But now these cells are highly pure, and so you can actually see that even highly related classes of neurons would express link RNAs, different classes of link RNAs. So this, I think, is a fundamental new level of regulation that we should study to understand how this diversity is actually generated.
Paola?
Yes.
Question here.
Yeah.
These link RNAs, are they related to the transcription factors or other genes that are close to them?
Right. So sometimes they sit just next to the gene that they regulate, but there are also cases of link RNA that are sitting very far away from a certain target that are also regulatory to that target. That logic is not quite understood, I have to say, because only now people are beginning to eliminate these link RNAs and really understanding what the heck they do, basically. But so both cases have been reported. So here's the link RNA. We mapped 5,000 new ones, of which about 800 here show incredible levels of specificity within individual classes of neurons. So there was at some point a theory that these link RNAs don't do anything. It's just a junk RNA or junk DNA, but it's impossible to think that you have something that is so specifically expressed in related neurons that doesn't do anything during development, in my opinion. So here is an example of a link RNA that is specifically expressed in the red cells, which are the subcerebral neurons. Here is an example of one expressed in the corticothalamic neurons, and this is another one expressed specifically in the callosal neurons, and these are just examples. And indeed, we have a paper that just came out together with John Green showing that if you knock in a LAC-Z reporter at the loci of this link RNA, and then you look what the LAC-Z is, which is a proxy for where the mRNA is expressed, you get this very interesting profile of distribution. For example, this link RNA, brain 1B, early on in development is quite nicely expressed in the MGE and in the LGE, and only very lightly in the CGE, and then later becomes expressed in the progenitors of the dorsal telencephalon and in the developing cortical plate, and acquires this beautiful arealization. Actually, you can see here the area distribution in S1 and V1, and not very much in M1, for example. And not to go too much into it, but if you knock this thing out, you have a reduction in the proliferation of all types of progenitors around the ventriculars, the ventricles. These are not all the progenitors, but both the ventral and the dorsal progenitors in the subventricular zone. And this results in the reduction in the production of upper layer neurons, as well as you can see here in the reduction in the somatosensory cortex and abnormal organization of the barrels. So there is a role that this link RNAs play, and it's a whole field of link RNAs now that are a way to be understood. We haven't looked. I have the mouse. I can give it, we're not going to, but yes, absolutely. And we have many more for which we have just profiled the distribution that are expressed actually in MGE and CG. And I would imagine that that could regulate the proliferation or the fate of those progenitors, absolutely. We should talk about it. So here is something that I feel kind of strong about. We have been profiling neurons for a very long time, we and others, not only us. And I have a feeling that every time this data is published, it's basically going to be deposited in some sort of NCBI data set that is never used again by anybody in the field, because the data that you find there is raw data. Unless you're a bioinformatician, you're never going to go get that data and analyze it yourself. And so we thought that maybe we can begin a trend of building easy-to-use data sets that are linked to websites. The first one of which we call DCON, for Developing Cortical Neuron Transcriptome Resource. So what this website is an easy-to-use interface where you go in, you type in your gene of interest, your neuron of interest, your molecular pathway of interest, and it will pull up the data of which I showed you just the tip of the iceberg that we generated in an analyzed form. You also can access the raw data if you are interested in analyzing it. But the idea here is that this kind of resources may become a coalescence point where people would deposit their own data as well or link it to it, so that the entire field can actually access the data without having to sort of reanalyze raw readings. So this is the address of this website. Go see it. Let me know if you like it or you don't. My hope is that we now did just transcriptome, but that we can combine epigenetics and genetic analysis of this neuronal subtype in the same place to take a systems approach rather than just look at one thing in one neuron type to understand how this whole diversity of cells is really generated. So I'd like to end the talk with a couple of slides, like snippets of how this approach that I just told you about can actually be applied to the purification of neurons in a variety of cellular contexts. And I'll just show you a couple of slides from these 3 points. First of all, these now can be used for the purification of induced neurons. And by induced neurons, I mean neurons that are generated either from the directed differentiation of pluripotent stem cells, by direct lineage reprogramming of other cell types in the dish, for example, or in vivo. You don't need genetic labels, so this could be an approach. It can be used for the purification in principle of human neuronal subtypes from either post-mortem samples that have been fixed and stored, material after all is fixed, or frozen. And I should say that this works with nuclei as well. So you can just access the data banks, the brain banks, and get a bunch of samples from there to analyze actually human neurons. And I think that this is important because we all know that a lot of diseases are due to death or degeneration of specific classes of neurons, yet we know nothing about why some classes of neurons are selectively affected by a certain genetic mutation that leads to a disease and others are not. So for the first time now you can take out the cells that are affected in a disease and really understand what's wrong with them from human samples. And then finally, for those of you who are interested in evolution, this allows the purification of cells also in organisms that, despite CRISPRs and all the genome modification approaches that exist today, are honestly not so easy to modify. And so let me tell you what we have done very briefly here, again, tip of the iceberg. So this is one approach that Marisa will tell you about in details, where one can create human cerebral organoids starting from induced pluripotent stem cells taken from anyone basically of us. And what you do is basically you take your induced pluripotent stem cells, which are obtained from directed reprogramming from skin, for example. Culture them in vitro. By day 10, you make these nice neuroectoderm spheroids, which then grow. And by day 18, they begin to make these bulges, which are basically, when you go and stain them, a beginning or a primordium, basically, of the neuroectoderm lining a ventricle that you see during normal human brain development. And then you take basically these organoids and you put them inside these spinning bioreactors, here shown in my incubator, and you spin them for months and months and months, and they grow and they continue to develop up to a point. Of course, there is a limit to what you can do here. Let's see. This is one of these organoids just at 30 days, so it's a very young organoid, but just to show you that it forms these ventricles. This looks like a lateral ventricles. This has been now infected with a virus that labels the progenitors, which have this sort of radial glia, or maybe you can say neuroectoderm type of morphology. And then you can grow them for much longer. This is a day 105. We're still spinning them for many, many months. And you can stain them for the same markers that label specific classes of projection neurons, for example, in the normal cortex. And it's hard for you to see here, but they form these beautiful sort of progenitor layers and a cortical plate-like structure. And within this structure, we begin to see upper layer-like neurons and deep layer-like neurons. In some of the organoids, we see interneurons migrating from ventral structures up to the cortex. So it's sort of recapitulating a process of cortical development. And now we can use these antibodies against this specific transcription factor to actually isolate specific classes of neurons from these organoids and profile them at a molecular level. You can make the organoids from, of course, patients with specific diseases, or from stem cells that be genetically modified to carry a mutation associated with a disease, and now you can profile human neurons that carry that mutation. This is an immunostaining of a coronal section of a human fetal brain at gestation week 16. And you can see that they have this beautiful cortical plate which stains again from some of the transcription factor that labels the different classes of neurons. Here are two examples of that. And what we have been able to do is to then take this fetal tissue, go through the procedure that I described to you, being able to really label these different classes of neuron inside this fetal developing cortex, put them through FACS sorting and separate the different populations. So one can begin to study really in high throughput neuronal diversity in the human developing cortex, and like cortex, any other part basically of the brain. And finally, this is turtle. So we are very interested in understanding what is the molecular evolution of neuronal diversity of the neurons that we have in the cortex. In order to do that, we need to be able to profile different classes of neurons in organisms that are not genetically modified. So this is Scripta, and what we did here was to stain for different markers that again label specific class of neuron. This is one example. You can see that it stains really nicely. And then take the dorsal cortex, reduce it to a single cell suspension, go through the procedure I just described to you, and being able to actually isolate for the first time from the cortex of a turtle different classes of cells. And so now you can do a cross comparison of molecular neuronal diversity, I should say, among different species during evolution, and not only during development of the same species. All right, so to conclude, To conclude, I think that pyramidal neuron diversity, and like pyramidal neuron, any other type of neurons of the central nervous system can be profiled in high throughput. So this is the key word here. In high throughput, quite easily without having to genetically label the cells. And this really opens the door to this system level approach, where you basically purify different classes of neurons from the same tissue at different stages of development through evolution. And you really co-analyze the data. You can also profile them at the transcriptional level, at the epigenetic level, at the proteonic level, and put all the data together to really understand how these identities are established. And this is, again, one example of a website where we put the data, and I would love to hear some feedback from you about it and whether you would like to link your own data from this website to be able to have things basically all together. This is us. I only told you today about some work done by Simona Lodato on the transcription factor FASF2. Emanuela in Georgia work on the organoids, and Brad, together with Loyal Goff in John Rin's lab, developed with us, help us develop the approach that I showed you for high purification of neuronal subtypes. So I guess I'll stop here, and I speak really quickly, and I'll take questions.
I have a question. Did you, with this method, did you try to see also whether you can purify the same kind of neuron population, but in that underwent different activity? So, like some transcription factor, the activity-dependent regulated, and you could pick somehow different levels of one transcription factor on that basis.
We haven't done that, but it can be done. I think. After having seen, the differences in levels of expression of this transcription factor that the methods can really detect, I think that can be done. The really key component here is how good your antibodies are. So not every antibody will perform as well. So don't take an antibody that barely gives you a signal in immuno. It has to be a decent antibody. And so what we're trying to do there is working with companies to try to actually generate a kit that would have a set of antibodies that work really well, that one could just use instead of going through many different antibodies. So Josh Sainz developed a similar method for the retina, very, very similar. I don't think he's published yet, but we talked a lot about it, and he tried many different antibodies that stain different types of retina cells, and many did. So as long as the antibody is good, and then you can combine as many as you want.